Hi Simon, so how you doing?
I'm good, thanks, Tim. How are you?
Yeah, not too bad. So today's guest is kind of a little bit like genetic, chemical engineering, sort of proteiny stuff that you do, but also like evolution and like how evolution works. And he envisages a bit of a brave new world, to be honest.
Yeah, absolutely. Sort of exciting, a little bit frightening, a little bit, a little bit of everything.
Yeah. So what for our listeners? What he thinks we're just, we've started as we've gone into a transition where people have now designed on the computer a virus. They've built it and it kind of works. It goes into cells and makes copies of itself. So they've done that from scratch. And he thinks this is kind of signaling an age where we can author life, we can author DNA, and we can build things. So we could we could whatever we could envision, we might be able to get biology to do it. And it's I mean, he admits we're not there yet, but he thinks we're there, and he talks about various uses for it, whether it's in medicine and what have you. But obviously, the kind of headline eye-catching stuff is we might be able to sort of build like Jurassic Park, but build them from scratch, recreate dinosaurs from first principles.
Yeah, I mean that's the really exciting and novel thing, right? So not just sort of tinkering with what's there already, but starting from a blank slate.
Yeah, so flying hippopotamus, maybe. Who knows? Something like that.
Is that the first thing your brain goes to, Tim?
That's always the first thing my brain goes to.
Yeah.
Flying hippopotamus. I think, terrifying. That's what would really terrify me.
Right, shall we get him on?
Yeah, let's call him in.
Yes, thank you for having me on the show. My name is Adrian Woolfson. As you can tell from my voice, I'm originally from England, but I've been living over here in the US for 15 years now. Most recently in San Francisco. My background is in medicine and molecular genetics. I trained in medicine in the UK, did about 7 or 8 years in the laboratory of molecular biology with Cesar Milstein, the guy who made monoclonal antibodies. Went into industry, BMS, Pfizer, head of R&D at the gene editing company called Sangamo, chief medical officer of a cancer vaccine company, and then ended up forming my own genome writing company. And most recently have written a book called On the Future of Species.
Which is fantastic. I really enjoyed reading, and that's out on February the 12th, I think, isn't it? It's Bloomsbury that's publishing it.
That's right. Yeah, that's right, Tim.
And available from all good independent booksellers, but also on Amazon. So, thoroughly recommend it to our readers. And the premise of the book is we're basically entering, or just in the process of entering, a really new age where we can author, we can design genomes, we can edit genomes to make super species or completely new species, things that evolution hasn't discovered before. Is that a fair overview of where you think the technology is at, or very soon to be, by combining gene editing and AI?
Yeah, I mean, basically, I think that we're at this kind of critical phase transition. Yeah, it's a key moment in the history of life. And actually, the purpose of my book is to call that moment because we've actually entered it. We're not about to enter it. We have entered it by stealth. And people need to know that. We're on the nursery slopes. We're not on the double diamond black runs. We're on the kind of 6-year-old ski bump, but we're on the slope, so to speak, right? And it's just a question of time before our skills mature enough to get us onto the double diamond runs, right? But the key premise of all of this is that for the last 4 billion years of life on Earth, all life has evolved by a process of evolution by natural selection. That process has no foresight, no intentionality, no guidance from an intelligence. And science has been a process of observation of received life, which is modified incrementally over time by a kind of this kind of sieve, if you like, a selection process, which operates by randomly changing DNA but there's no actual oversight or design. And the key transition, if you like, is that all of a sudden, through the use of AI and the ability to write genomes at scale, suddenly we acquire the potential to write biology, to author it, to design it with intentionality. So all of a sudden, evolution isn't the only game in town. There are now two authors. There's evolution and there's design driven by humans guiding AI, a kind of collaboration, if you like, between natural and artificial intelligence.
So it's a bizarre kind of, I mean, I'm not now religious, but I come from a religious background, and it's almost like a reversal, right? It's of the kind of traditional religious way of thinking where there's a designer, and then so this is where we've had evolution comes first, and then the sort of designers are coming along secondary.
Yeah, I mean, obviously, if you're able to design life, and as I said, we're on the nursery slopes. A chap called Brian He at the Arc Institute in Stanford has technically used an AI-driven process to design the first ever entirely new and synthetic species. It's actually a biological entity because it's a virus, not a more complex organism, but nevertheless, it is a designed, AI-driven designed genome, right? So that's a kind of proof of concept. We can't write new types of zebras or giraffes or whales just yet, right? But what it shows is that the design process underlying all life is rules-based. And if we can unpick those rules one by one, we will eventually have the ability to build life as we desire. Right now, is that, I mean, I think you're alluding to the questions, are we playing God? Have we acquired some kind of divine potential or capability? And my answer to that is what we've acquired is responsibility, right? Because before all of this, you know, we just passively tinkered with what was out there. You know, gene editing, for example, is a bit like redlining a text. You know, you can tinker with genomes, but you're fundamentally constrained by what nature has handed you, right? You can't really deviate from the tree of life very far, right?
Sorry, go on.
I was just going to say, with the ability to write life and design it, we can actually explore potentially the forest of life, which is everything which has never been, which is far, far greater than what has been. But also we can re-examine what exists and think about how we might wish to change it for the benefit of society and medicine.
So the nursery slopes, I'm just sort of thinking what they look like at the moment, where we are. And we had one guest on the podcast a few months ago, a guy called David Baker, who won the Nobel Prize for Chemistry. And what he did was he basically designed a new protein which would bind to snake antivenom. He then took the genetic code to produce that new protein, put it into bacteria, produce the protein. So it was a bacteria that he was modifying. But then when he injected that protein into mice, he discovered that it did actually work and it neutralized the venom. Is that the sort of, when you talk about the nursery slopes, is that the sort of thing you're thinking about?
Yeah, that's sort of extremely consequential and kind of on the ski lift, if you like. It's because, I mean, and that's not to demean what David's done. David's a genius, as is John Jumper, Demis Hassabis, all of those guys, because what they've done is they've shown that in the same way that mathematics was the language of physics, it seems as if the language of biology is best interpreted not using mathematics, but using AI. And what they showed is that if you use AI simply to look at the simplest kind of functional units of biology, which are genes, protein encoding genes, you can actually, by interrogating huge data sets, pull out principles which we don't actually even understand, which is, I didn't really finish the question about why we're not gods, but we don't understand the rules, right? And we don't make universes, by the way, either, right? So we're very, very far from any type of metaphysical power. But as I said, the key is we've acquired responsibility. But what David has done, David Baker, and David is a genius, as is Dennis Fasibis, as is John Jumper. What they did is they've kind of used AI to enable us to predict the structure of proteins and to redesign proteins. And proteins are kind of atomic units from which organisms are built. What they haven't done is go beyond that and actually kind of generalize those principles to whole genomes. And the person who's leading that kind of particular stream of scientific endeavor, which is to me what it means to be on the slopes. That's why I said they're on the ski lift. Right? I mean, and that's in absolutely no way to trivialize what they've done. Because what they did laid the foundations for everything else that goes beyond it. I mean, it was absolutely of seminal importance, and they all were very worthy of their Nobel prizes, actually. But actually, in my view, the next step is the one that's really kind of takes it to the next level, which is to take those same kind of approaches of interrogating huge databases using AI and then technologies for writing DNA to actually build genomes, right? And so Brian He, as I mentioned before, who I think is really the pioneer of this field, recently showed that he could use AI interrogating huge databases of viral genomes and bacterial genomes to design a new viral species by the formal criteria. So that, to me, is the proof of concept that AI can design new life. It's like, to me, it's like a Wright brothers moment. The plane has kind of bumped off the ground. It hasn't quite taken off, but we know that we can fly in principle. And it's just so, a matter of time before we build a 747, right? And I won't try and guess how long that will be, right? But we have taken off, right? This is happening. People need to know about it. There are obviously huge societal consequences. We need to ensure that there are guardrails in place, that there's an alignment internationally about what we should and shouldn't do. We need to do this wisely, safely, responsibly, in an equitable manner that benefits all humankind and that benefits society in a meaningful and safe manner.
But Adrian, isn't the big impediment to sort of doing that still this big gap we have in the genotype phenotype relationship?
Yes, you're absolutely right, Syma, and that's where we believe AI will help us. And AI, remember, it's still in its infancy. Really, the type of AI that's enabled us to, people like David Baker to do what they have done is, it's only a couple of years old, and that was limited by technology. It needed these computing architectures called GPUs, right, which were developed for the gaming industry, actually, driven by arcade games like Space Invaders. I remember that. I hope none of my old school teachers are listening, or they may not even be alive. But I confess on this program to missing the odd day of school to go and play Space Invaders in a cafe in Hampstead. But in fact, the graphics enabled which at the time was a cutting edge video game, were exactly the same graphics that had been used for AI, and it just turned out that they were useful for AI. And that's what's enabling this new revolution. Now, we're not going to be able to understand the rules of life in a day, right? We're doing it incrementally. And that's why I said, we're on the nursery. So whereas the rules, and they're grammatical rules, because biology is a language, we're talking about the language of life. But instead of trying to learn German on day one of the most complex languages grammatically, we're starting off with Pidgin English, which is a viral genome. Or if you want to think of it in terms of authorship, one of the most memorable books of my childhood was Topsy and Tim Go Fishing, which is the most intellectually taxing book today for us, right? But it was at that age, right? But it's not Dostoevsky, right? Or War and Peace, right? But it is on the road. Once you can write Topsy and Tim Go Fishing, then you're on the road to reading and writing Dostoevsky.
Perhaps for our listeners, we should just explain what we mean by that gap between just thinking between genotype and phenotype. So what we're saying here is just because we know what a genome is, we don't know what the sort of consequences of those genes are, how they manifest in an organism, how that organism interacts with.
One other important point there, Syma, and this actually goes back. I've just been reading. I shouldn't be plugging someone else's book when I'm doing, but I just read a brilliant book by Gregory Radek, which is about the sort of history of Mendelism and how actually, very few genes are Mendelian actually at the end of the day, and that's because in reality, there are actually multiple layers of information that contribute to what used to be called traits or phenotypes in the modern parlance, right? And actually in my book, so I'll go back to my book, I actually use a term which I think is very useful. Nobody else has used it. And I refer to the informiome, right? And the informiome is the information that constructs a phenotype. And that information, it's true, is grounded in DNA. And I'm not saying genes only, because genes, protein encoding genes, are just a small part of the story. Most of the genome transcribes RNA molecules, and they've been overlooked historically, right? And we've talked about there being 20,000 genes. If you account for all the RNA producing genes, there are many, many more, right? However, it's not just genetics which controls phenotype, it's development, learning, culture, the bugs that live within you, the bacterial genes in your body far outnumber our own. And it's now been clearly shown that having one species, more or less, of bacteria in your body can influence your response to immunotherapy. It can switch other genes on and off, right? So the genome will never be the whole story. And therein may lie the fundamental problem, that we may not ever be fully computable, even in principle, right? Because in order to fully describe a human being or any other organism, you would need a complete description of all of these different levels of information, which may not be possible even in principle, although a genomic description may be the closest we get.
So I mean, given that, what do you envisage the starting point being? So do you imagine are you starting with you want to make a new species? Are you starting with a new species of bacteria, let's say, so still something reasonably simple compared to, say, a multicellular organism like you and me with 30 trillion plus cells? Are you envisaging we start with a blank slate, or are you saying, okay, well, we know that there are some highly conserved genes that all of life uses, we'll throw a few of those in, and then we're going to start sort of saying, okay, I want it to have green hair, the dashing good looks of Tim, whatever. How are you envisaging this? What is it? The blank slate?
Well, I think the first thing to say is, why might we wish to do this, right? My prediction, and I think I'm definitely right about this, is that we're entering the age of biology, or what I call generative biology, what people in the UK generally call engineering biology, right? But basically, biology is set to become infrastructure. That's really important. So everything that's done through destructive methods like chemistry could in principle be done using biology, which is sustainable. And in order to do that, we need to turn biology into a programmable material like computer code, right? And into a predictive engineering material. So let me give you some examples. For example, you could store most of the world's information, and storing information, as you know, is incredibly energy consuming. You could store most of the world's information in relatively small amounts of DNA, right? So DNA could be used to archive information really efficiently. You can use DNA to make biomaterials. I went to the Biennale in Venice the year before last, and they were building bricks from mushroom proteins, for example. People are making car seats out of biomaterials. Spider silk has the tensile strength of steel, right? People are making bioconcrete, which repairs itself, right? Bioenergy, biocomputing, right? And then another huge, so it's not just, making new species is part of the story, but the bigger story, if you like, is just making biology programmable. And also enabling us to really leverage AI, because at the moment there's a huge disconnect between our ability to design things on computers and our ability to then build those things and then feed back to the AI. And in order to do that, you need to be able to build DNA at scale, something that I've actually been working on with my colleague at Caltech. And you might see something in the news fairly soon about that, because it was the bottleneck. You need to be able to design DNA using AI, but you also need to be able to build DNA at scale, rapidly, efficiently, accurately, at scale, and at really low cost, right? So to go back to your question, I just wanted to sort of contextualize that, right? I think what really makes this difference to CRISPR was a huge revolution, let's face it. Jennifer Doudna, all of those guys, George Church, CRISPR allowed us for the first time in history to deliberately and specifically change genomes in particular positions in a way that breeding could never do. Breeding was a sort of black box. We could kind of crudely guide phenotypes, traits, in certain directions. And in fact, corn is a good example of the result of that, because corn started off, as you know, as Tisonite, which was a wild kind of weed, actually. And through breeding, selective breeding, we created a global cash crop, right? And so it can be done, but it takes, I think that took 30,000 years, right? Something that could have been achieved today in a couple of days. So gene editing is great, and it was an unprecedented skill. And I spent, several years of my life working on gene editing. However, gene editing, as I said earlier, is like redlining a text. You are an editor, right? And Tim, you've written a book, I've written a book, right? We know that we pride ourselves because we are authors, right? Authors don't edit, so when you wrote your book, Tim, you didn't go and get, Macbeth and start turning it into a book on evolutionary biology or the history of the universe, right? You just sat down with a blank page and wrote out the contents of your mind in a totally unconstrained manner, and you allowed your creativity to run in any direction that you wanted it to do. And that's the difference between editing life, either through selection, which is one way of editing, or natural selection, or through literally gene editing. When you author life and design life using AI, using rules which you may not fully understand, in fact, we don't understand them at all, but we can discern them, then that's a totally different ballgame, because now we don't need to be constrained by history. In fact, quite the opposite. Life can break off from the tree of life and become ahistorical. And in fact, we don't need to reference existing life in any shape or form. We might get some clues from it as to what works and what doesn't, but we're no longer constrained. And remember that life as we know it, both past, present, and future, is but a tiny grain of sand on a massive beach of possible life, endless beach. So we've explored the tiniest fraction of possibility. So just imagine what's out there. Now, just by way of example, imagine that a dodo had never existed, right? We'd have no idea that a dodo-like bird exists in principle. In fact, a dodo has a timeless mathematical existence. It's extinct, but the description of a dodo mathematically, biologically, will always be there in perpetuity for the rest of time. And similarly, it would be possible to build a map of all possibility one day using AI, and to be able to, it's like the map of the world, really, but it's much, much bigger, obviously. And over there, you've got flaminoceroses, a cross between a flamingo and a rhinoceros.
But Adrian, isn't this really scary, what you're saying, in the sense that the potential for disaster seems to me to be enormous. We could create,
as I said earlier, I think it's clearly a technology which is very consequential. In the wrong hands and without a clear moral kind of foundation, and with the kind of values that I alluded to earlier, a sense of responsibility, doing it wisely, and really thinking deeply about how everybody could benefit from it, how society can benefit, then, without that kind of consensus and guardrails, yes, it is dangerous. But like all technologies, this is a dual-use technology. So there was, think of the wheel. What a dangerous technology, right? You can use it to make a wheelbarrow to take your fruits and vegetables into market, or you can make a chariot, a war chariot. Look at fire. That's an incredibly dangerous dual-use technology.
But Adrian, but the big difference is
down the city.
Sure. The big difference, I'm going to push you on this a little bit. The big difference here, though, with the wheel is I can anticipate both uses. Which
you've already anticipated the dangers of building an amp the size of the Empire State Building, although we are limited by physics and chemistry, so I'm not saying that's possible, right? But look, here's the thing, right? As I said, the reason I wrote my book is not to proselytize about this or even to advocate for it. It's happened. I'm calling a moment. I want everybody to know this has happened, right? So it's not a question of should we do it. We have done it, and it continues to happen at an exponential rate. The ship has left the dock, right? So what matters now is that people know it's happened, know enough about it to have an opinion, and that there is some kind of framework for a global consensus to make sure this happens in a responsible and thoughtful manner for the greater good of humankind, right? And there are many, many good uses this type of information could be put to, not least human health, right? We haven't really talked about that. Yeah. However, clearly in the wrong hands, this is a very dangerous technology as well.
So AI is central to what you're talking about, machine learning and what have you. And it obviously AlphaFold, which was one of the breakthrough uses of AI. It was able to work out how strings of amino acids would fold into proteins in many cases. It needed a big data set to learn from, where we had the 3D structures of proteins and we had the DNA sequences in order to do that. Do we have anywhere near enough data at the moment to do anything beyond a virus? And the reason I ask that is for our listeners, viruses obviously use the host cell to replicate. But we've got also, if I just think about building even just a simple cell, you've got potentially gene cytoplasm incompatibilities that we don't understand. Yeah. And that's just one thing. There's a huge number of things.
So many thoughts come to mind when you say that, and I wish I could give you 10 answers simultaneously. The first thing I like to say is that, quote Peter Medawar, science is the art of the soluble, right? So I've absolutely no doubt that we'll be able to compute all of those things, right? And the ability to write DNA in a manner that matches our ability to design will enable us to get that feedback as we design to learn those rules, right? I just want to quickly, though, before I sort of expound on that, but just go back to AlphaFold, because it's a really important thing for the listeners, and some of them may not know what it is, but it also illustrates a really good point, right? So just to go back to that, so AlphaFold was an AI-based method for predicting how a one-dimensional string of amino acids folds, because proteins fold like a bed sheet or whatever, in three-dimensional space to form a structure. And ultimately, biology relies on structure, right? So proteins have to form a three-dimensional structure. Now, for many, many years, led by actually initially a former colleague of mine, Alan Furst in Cambridge, people tried to understand the rules, the physical and chemical rules that took you from a one-dimensional amino acid sequence to a folded protein. There was actually a competition every year to see, people were given one-dimensional sequence, saying, can you predict how this will fold? And then they'd compare it to a structure that had been derived crystallographically, which is how traditionally, the structures of proteins have been determined by firing X-rays at them and then solving the structure, which takes about 6 years generally, right? And really, the whole field of prediction was going nowhere, right? But trying to use physics and chemistry. So along comes John Jumper, Demis Hassabis, and so on, and people like David Baker. And they start to interrogate databases, matching the known database of structures with sequence, And lo and behold, AI works it out, right? Now, interestingly, how did it work it out? We don't know, right? Because
Well,
AI did not understand all of the rules. All it did is it
yeah.
saw the patterns and uses those
And,
patterns to make the mapping, right? So,
and that's why I'm asking the
yeah,
question about, do we have enough pattern out there for the way that
yeah.
even a
And
bacteria works?
so
Yeah.
coming back to that, the answer is right now, probably not, right? We do actually for viruses and bacteria, there's a huge database of viral and bacterial sequences. Fortunately, as Jacob once said, that the rules for building the rules of building an ant are the same for those of building a human, or something like that. In other words, the rules that I think I definitely got that wrong, but the principle's the same, right? In other words, biology uses a universal set of construction rules, a kind of universal grammar. Some of you readers will remember Noam Chomsky, who talked about a universal grammar which underlies all human languages, right? It's the kind of potential for language, right? And I think, it's probably almost certainly the case that there is a deeper universal grammar of biology which underlies the individual languages, the individual grammars of every species. So the grammar of tortoise-ese, the language to build a tortoise, right? It's not the same as the languages of leopardese, the language to build a leopard, but they share a lot of similarities, right? So if you learn how to write a tortoise, there's a damn good chance that you'll eventually learn how to write a leopard, right? But hey, like I said, we don't start with Dostoevsky, we start with Topsy and Tim, or something even simpler, right? And once you learn how to read Topsy and Tim, and well, read and write, right? Once you can write Topsy and Tim, is where we are talking about authorship. And remember, and just important point I missed earlier, that we started off observing biology. We finally learned how to read biology when Fred Sanger in my old laboratory in Cambridge worked out how to sequence DNA. We then learned how to edit DNA, thanks to people like Jennifer Doudna. But hey, we're at a totally new moment. This is a phase change. We now are learning how to author life, like literally write life creatively with intention. In other words, we've transitioned from being monks in a scriptorium, blindly copying ancient texts, making the odd mistake every 2,000 letters, I think the monks made, right? To more like a kind of Gutenberg situation. Gutenberg printed the first Bible in 1455 or something like that, right? And this was the sort of industrialization of books, really, right? And suddenly, this industrialization enabled the everyman to start being creative and writing their own books. You didn't have to just copy the Greek masters. You could now. It was cheap enough and easy enough that anybody could have an idea and create a book, right? And that was basically the basis for both the European and in specifically the Italian Renaissance, right? So I think we're approaching this Renaissance moment in biology where fairly soon we might find, that your 8-year-old kid is designing a new organism on their computer. And I think we've all got views on whether that's a good idea or not. But actually, this is, I think biology will become programmable, just like kids sit at home programming video games. They're going to be programming life pretty soon. And I remember when I was a graduate student, with Cesar, my old boss, who invented monoclonal antibodies, won the Nobel Prize, and 10 years later, there was still only one approved monoclonal antibody, right? And I remember there were all these really mean articles in the press that monoclonal antibodies were a failure, they weren't going anywhere, it was hype. It just took a little bit longer than people thought, right? Yeah, it's going to happen. It may take 2 years, it may take 5. We don't really know. It's going to happen, right? Better that we're prepared for it. My book was meant to be a kind of, I don't know if you ever came across the Baedekers. They're this sort of 19th century travel guides. They were the first kind of travel guides for independent travelers, which had tips on, you know, what hotels to stay in and the best sites to visit and all of that. And I kind of, when I was writing my book, I saw my book as a kind of Baedeker, you know, kind of a travel guide, but also a kind of trusted companion that you could put in your coat pocket as you navigated the future of life, and use it as a kind of book to guide you and inform you. And that's how I hope my book will be perceived. It's definitely not proselytizing. It's definitely not advocating. It's simply saying, here it is. This has happened. Yeah. You better know about it.
And you do that. You do that very well. We're running out of time. Syma, a question from you, and then I've got one last question, and then we probably should wrap it up.
I've got quite a sort of a out there bonkers question. Okay, you get to design a tiny little organism. What's your organism going to be?
What you mean? What's my pet organism?
Yeah.
What will it do?
We've already. I mean, we are already doing that. You know. So let me tell you what we're actively working on, because I've written about this, but I'm also doing it on a day-to-day basis. So, and you'll both be aware of this. Like, you may even have had somebody on your program talking about it. But it turns out that bacteriophage, which are the viruses that prey on bacteria, turn out to be actually an alternative form of antibiotic, right? And these have been used actually quite extensively in the Ukraine war, because a lot of the soldiers there have got multiply resistant Staph aureus infections, and they've shown actually they can be treated pretty effectively with bacteriophage, right? So, you know, one can imagine a situation, particularly in this age of global antibiotic resistance, multiply resistant organisms, which is really becoming a major global problem. And as you know, very few pharmaceutical companies see much commercial incentive for working on antibiotics compared to, say, anti-cancer drugs. And, you know, we can easily imagine a situation where we can determine what infection somebody has, and then, you know, press a button and in a bespoke manner spit out a bacteriophage sequence that will kill that bacterium. That's a genome, right?
Yeah.
And bacteriophage do not affect human cells at all. So totally safe, right? So that's a really simple, practical example of today of how we can use this technology. Let me give you another example of something.
Well, we're kind of running out of time. So give us an example of something a bit more complicated than a virus.
Yeah, I'll do that. So we're working with a group in Cambridge right now to build what we call a neochromosome, which is an artificial chromosome, which can be used to reprogram the genomes of crops, for example, to make them drought resistant or pest resistant, or require, you know, different low nutrient, they can survive in low nutrient conditions or whatever. In other words, moving from editing, which is very incremental, to actually reprogramming the genomes. And this could hopefully form the basis of a second green revolution, because we're going to need to be able to sustain all of the new humans coming into the world at a kind of exponential rate, and in a way that doesn't destroy the last remaining wilderness and pollute the world with chemicals and so on and so forth, right? So this is a very immediate utility of this technology.
So do futurists tend to get it right, people who are predicting the future, do you think?
You know, I've always thought, Tim, right, that the human imagination has this incredible ability to predict the future, right? And, you know, we've always talked about things like time travel, and who knows if that's possible. There may be some things which aren't possible. So I'm not saying that anything you can imagine is possible, but I definitely think that many of the things that we can predict are possible. The best example of this was obviously Leonardo da Vinci, as we all know, you know, predicting the existence of helicopters and so on and so forth. Francis Bacon anticipated synthetic biology many, many years ago. He died in Highgate in London when he got out of his carriage in the cold to do an ad hoc experiment on a chicken. But he actually wrote a book after being released from the Tower of London, in which he actually imagined that nature could be engineered. You know, this was years before we even knew what a gene was. So his imagination was able to fly into the future and, you know, imagine different things. Are those alternative worlds better? Are they natural? Are they unnatural? who knows, right? I don't think that the one defining thing about humanity and about nature is that it's in a state of perpetual change.
Yeah. Well, look, I wish we could keep talking for more because it really is a fascinating subject, and I really recommend your book for to our readers because I enjoyed reading it, and it is very thought provoking. And you're right. There's as we move from the from the nursery slopes, there's probably going to be a lot of falls, maybe the odd broken leg, and even the odd lost ski, I suspect. But
Yeah.
I'd recommend it. On the Future of Species, Authoring Life by Means of Artificial Biological Intelligence is coming out in February the 12th, published by Bloomsbury. It's really thought-provoking, very knowledgeable, and really enjoyable read. So thank you very much for joining us, Adrian. It's been fascinating.
And thank you, Tim and Syma. It's been a pleasure talking to you, and thank you for your kind words.
What did you make of that then, Simon?
Oh, it's fascinating and a lot of fun. And I think, as I said at the start, I won't lie and I'll say that I find it exciting, but also a little bit scary.
No, it's true, and I mean, I think we probably, we tend to think of like the real extremes, which we're a long way off. But I think some of his points about being able to design, for example, viruses or phages that will kill particular types of bacteria. So, a treatment for health. I think those are the first things, and potentially really, really positive. But of course, if you design a virus, there are also bad viruses out there. So that's where the kind of ethics stuff that he says we need to think very carefully. He touches all on all of that in his book, in fairness. We didn't get time to discuss it.
No, no, no. And I think, just sort of thinking about it after, after we've interviewed him, it's a little bit like we're going to all have these sort of almost biological 3D printers, right?
Yeah. and and I mean, I think, it's interesting, isn't it? Because we had on Omar Yagi, who's designed these new compounds, which are coming online there. David Baker, who designed the protein and won the Nobel Prize for it. The idea of sort of authoring complex organisms, at least beyond viruses, I suspect is some way off still because it's so complicated. You're
Yeah.
talking, uh, you know, you know, billions and billions of interacting molecules within each cell, and actually, yes, they do follow rules. There are chemical rules, there are physical rules, there are biological rules, and what have you. And we're only scraping the surface. So I think it's a while off, would be my suspicion.
Yeah, I think so, because these having the different components and then putting them together makes it more than the sum of the parts.
That's right. And I mean, I think he's true. I think he's right that artificial intelligence, much as it has, created things like AlphaFold we talked about, but also been able to, convincingly reply to us in language that we understand. That's why it's so convincing in that sense. It's come a long way, but and he's probably right that eventually we might get to this point. But I think artificial intelligence itself and machine learning has got to come a long way before we're actually there.
Yeah, I think so, because when you asked him about the sort of are the patterns there, are there enough patterns for the AI to pick up on? They could be there, but the AI, they have to be there in a way that the AI can find and use and manipulate,
That's right. And going from a genome, putting in a genome to just the shape of an organism, how many limbs it's got and what have you, isn't getting you there, right? Because you've got the whole developmental processes. You've got, we don't even really have not everyone can agree on the description of life, let alone, but really fascinating stuff. And I mean, such enthusiasm as well.
Absolutely. And I think for our listeners, if you have any questions or, comments, and as always, please write in, please comment on the various links on the different platforms you use to watch or listen. We'd love to hear your feedback.
No, and no, and a good book and worth reading. I recommend it. So I enjoyed it.
Excellent.
Cool.
Well, that's it for another week, I guess, Tim. I will see you hopefully at some point soon.
Yeah, no, definitely. And I think we should give a shout out to our producer, Rich, who's had a half a knee replaced. So he's on the mend. And let's wish him the best for a quick recovery.
as we said, he's training for Spurs soon. So, you know.
Ha ha ha. Okay, listeners. Okay, Syma. See you soon.
Bye.